Alrighty, guys, here we go with transcription. Third set of notes for the unit. So we've done the history of DNA with the scientists, then we did replication. Now we're moving into actual protein synthesis. Okay, so transcription is the first step for that, and it's the conversion from DNA into RNA. So this term, the central dogma, which encompasses all of the protein synthesis models, was actually developed by that guy up in the corner there, which is Francis Crick. So from the famous Watson and Crick. So Crick contributed quite a few things to the area of DNA and genetics in general. So the central dogma is the flow of the genetic information in a cell. And so how do we move that information from DNA to proteins is what we're going to be looking at. So you saw this diagram yesterday. And remember that when we're making proteins and we're talking about, anything having to do with making a protein, it's this right here. And that replication does not have to do with making a protein, right? That's just DNA copying into DNA so that it can be passed on, okay? And yeah, DNA gets the glory, but the proteins do all the work, right? So a lot of people focus on DNA in like crime shows and stuff because you can identify people with the DNA. But for our bodies, it's really the proteins that do a lot of the work, as well as RNA as well. So if we look at just a couple diagrams here, I'll point out a few things. So here's your DNA to RNA to protein pathway, and we're going to have DNA and we're going to do transcription and we're going to get an mRNA out of that. And then translation is going to give us the protein down here. This little wadded up ball of amino acids is our protein. And if we look over here, it gives us a few clues as well. If you look, here's the nucleus, and the nucleus is this area shaded up in green up in the upper left-hand corner. And so remember, DNA always stays in the nucleus, always, never ever leaves, right? RNA can leave, mRNA can leave, and that's showing this, right? So transcription is going to happen in the nucleus, and that's what we're going to focus on today. And then eventually we'll talk about translation, and that's going to happen out in the cytoplasm at a ribosome. Okay, so those are kind of the locations of where things are going to start taking place. So we'll come back to a whole lot of diagrams like this one down here over the next couple days. So metabolism is what really taught us about genes and how things worked. So seeing things come up that weren't right and working backwards and trying to figure out what went wrong showed us that genes were there and that they are really, when things don't go right, that's really what causes what we call a phenotype, right? So each disease is a phenotype, right? If somebody has a particular disease, then that's a phenotype. We can see that. And so this suggested that genes coded for enzymes, and when things didn't go right and enzymes weren't there and there was a lack of an enzyme, then you get a problem. And you might get Tay-Sachs, or you might get phenylketonuria, or you might get albinism, or one of these things. So if we look at our pathway down here, A to E, we have enzyme 1 that governs from A to B, and we have enzyme 2 that governs from B to C, and so on and so forth, right? And at any one of these, if you do not get, if you don't get enzyme 1, then you'd get the disease above B. And if you don't get enzyme 2, then you'll get the disease above C. So it just kind of, at any one of these, you could have things go wrong, and then you get some sort of phenotype or some sort of disorder. So am I just the sum of my proteins? Kind of, but not too important of a question there. But your genes are really dictating all the proteins that are being made, and hopefully they work correctly. And if they do, then you should be good to go. So these guys, Beadle and Tatum, couple more scientists for you, came up with the one gene, one enzyme hypothesis, meaning for every gene on DNA, it would make one enzyme, and that enzyme would be responsible for a process. And so, do you guys have any idea how many genes there are? Stop and ask yourself real quick, how many genes do you think are in the human body? So when I worked on the genome project, the notion was that there would be about 100,000 genes. When we started the project, they expected us to find about 100,000 genes that govern everything in our bodies. And the project went on for a few years, and they're like, it's probably not that many. It's probably 50 to 75,000. And the project kept going a few more years, and they're like, yeah, we're not finding that many. We think it's going to be about 30. And honestly, it ended up being somewhere around 20, I believe, give or take, when they were done. And that's really what governs everything about our bodies. So these guys won the Nobel Prize for the discovery that genes act by regulating definite chemical events. Okay. And we're going to mention something a little bit later. So I want you to remember this slide, one gene, one enzyme hypothesis. I want you to remember that, and I'm going to come back to it toward the end here. All right, transcription, copying DNA into RNA. Here we go. So we have to talk about RNA, right, when we get to making RNAs. And so ribose is the sugar. The nitrogenous bases are uracil instead of thymine, right? So notice right here, it should be, or normally is, an A with a T, but now A is going to pair with a U in RNA. So if you ever see a sequence of letters and it has the letter U, you know you're looking at RNA. If it has the letter T, then you know you're looking at DNA. Okay? RNA is single-stranded for the most part. There is actually double-stranded RNA, and there is actually single-stranded DNA. They don't really tell you this until you get to college or grad school, but those things do exist, and I'm going to tell you about a couple of them down the road in another set of notes later this unit that we'll get to. There are lots of RNAs. You guys probably know about mRNA, messenger RNA, and tRNA is transfer RNA, and rRNA is ribosomal RNA, and these are the three that you learn about in regular bio. siRNA we will talk about a little bit later, and there's actually a few others that will show up, and we'll talk about some of those as we get down the line here a little bit. So the steps of transcription, remember the other day I told you that these steps would keep coming up: initiation, elongation, and termination. Those were the same three steps we saw in replication. Now this stuff was slightly different. The stuff behind the dash was a little bit different. For transcription, we're just talking about preparing for copying into the RNA, the actual making of the RNA, the elongation part, and then the release of the RNA, as well as some editing that has to go on. So we will get to all of this. Here we go. So we have an RNA polymerase. So remember, in replication, we talked about a DNA polymerase, right? Replication had a DNA polymerase. So these are one of those things I was telling you about. Make sure you know that DNA replication or DNA polymerase goes with replication because you're making DNA. RNA polymerase goes with transcription because you're making RNA. Okay, so make sure you're up to speed with what words and terms and enzymes go with which process. Okay, so the RNA polymerase is going to attach to something called a promoter. Okay, and here's the promoter right here, and you can see this bracket right here shows you where the promoter is. Okay, and at the beginning of that promoter is something called the Tata box. Okay, and if you notice right here, there are letters, pretty bad arrow, but there are letters that spell out TATA, and we call that the Tata box, and that is recognized by RNA polymerase and by what we call transcription factors. Okay, transcription factors are proteins, and they come and they bind with the Tata box, and that is going to show RNA polymerase where to start. Okay, so this is how it knows where to start, because when you think about it, the chromosome is massively long, right? But genes are only, they could be really short and just be a few letters of code long, or they could be a few thousand letters of code long. And so you have to tell the RNA polymerase where to start to make the RNA. So this clear looking guitar pick looking thing right here is your RNA polymerase, and it's going to be the enzyme that binds and then goes on to make the RNA. Prokaryotes, RNA binds to the the RNA polymerase binds to the promoter directly. So no transcription factors on a prokaryote. Okay, no transcription factors. Eukaryotes need transcription factors. They have to have them, or they won't be able to make an RNA. Okay, so that's where to start. That's how it recognizes it. So a little bit more on that blown up picture here. We see the RNA polymerase, we see the transcription factors. So those are a suite of proteins which bind to the DNA and basically just are the big red arrow saying start here. Hormones can also bind to receptors, and they can either activate or suppress transcription, meaning it can be turned off or turned on, kind of like a light switch. Okay, so transcription needs to be controlled because you don't want to make too much protein. You don't want to spend all the energy making a bunch of protein you don't need. And you know, so you want to be able to turn it off, but then you have to be able to flip the switch back on and start making it when you need it. So we are going to talk about regulation in another set of notes, but just so you know that transcription can be regulated and it can be turned on or turned off, and that's what we call gene expression. Okay, whether the gene is being expressed. If the gene is being expressed, then you're getting the protein. If the gene is not being expressed, then no protein is being made. Okay? So choose your polymerase, right? There are 3 RNA polymerases, and you can see right here there is RNA polymerase 1, which has to do with rRNA genes, RNA polymerase 2, which has to do with mRNA genes, and this one in red right here, number 2, is the one we will focus on the most because that is the mRNA. And then RNA polymerase 3 transcribes the tRNA genes, okay? So each has a specific promoter sequence that it would recognize and know where to bind and where to start. Elongation, making the mRNA. So this is the nuts and bolts of how it gets made. So the transcribed DNA strand is the template strand, and the untranscribed DNA strand is the coding strand, which ends up being the same sequence as the RNA. I always find this to be a really weird name because they call it the coding strand, which to me, like that's the code you want, but it's the untranscribed strand. It's not the one that's used. It's the template strand. So if we look down here at the diagram down below here, you have the helix getting unwound a little bit, and then you have this box keeps, well, if it'll kick me out. Yeah, it kicked me out. That box just kind of keeps getting in my way every time I move my cursor and get a marker down there. But you have, again, we can only add to the 3' direction. Okay, so mRNA, here's your 5, and here's your 3. And so it has to be built in this direction, exactly the same as replication. You can only build 5 to 3. Okay, so in this case, the unwinding is going this way. Okay, let me get our terms back up here real quick. So the template and the coding. So the unwinding is going that way. We're building the strands this way. The RNA polymerase is adding the bases like this in this direction, right? This is how they're going to get added as we go to the right in this case. And that's pretty much it for now. Look, the coding strand is not the one being copied. It's the template strand down here, okay? So we'll keep going over this every in the slides here. There'll be a little bit more. So you have the RNA polymerase as shown in this diagram, building 5 to 3, adding in the bases, complementary base pairing. Notice we have the U that's getting put here with the A, right? But keep in mind, you still have T's. You still have T's on the DNA side, so A's are going to go in there, right? Not U's, okay? T doesn't pair with U. T is still going to pair with A. So, you know, if you look here, actually in the first one right here, you still have A's in the RNA. You just can't have a T. So whenever you feel like putting a T, just make sure you put a U there to go with that A, okay? 5' to 3'. And then termination. When you get to the end, when RNA polymerase reaches another sequence, it will have another code, kind of like the Tata box, but there'll be a termination sequence. It will recognize, it will stop, and then it will dissociate. The polymerase will release from the DNA, and it will also release the RNA transcript. And now we have an RNA transcript, and our DNA goes back together as a double-stranded molecule. Okay, and the RNA polymerase down here is now gone. It has come off. We made an RNA, so now what? So there are many types of RNA for different jobs. I think I mentioned those a second ago. mRNA is the messenger. That's the RNA that's going to carry the information to the ribosome so we can make a protein. rRNA is part of the ribosome. Ribosomes are made of rRNA and protein, so it's part of that substance that makes up a ribosome. And then the tRNA is the transfer RNA, and it's going to bring the amino acids to the ribosomes during translation. So tRNA will play a much bigger role in the notes tomorrow. There are other types that will show up in subsequent lectures. So a little bit about eukaryotes and prokaryotes. So in eukaryotes only, the mRNA transcript is extensively modified. It's extensively modified and processed before leaving the nucleus. We make the RNA, but then changes get made to it before it ever leaves, okay? So you can see, here's the eukaryotic cell on this side, here's the DNA, and you'll see this diagram refers to it as a pre-mRNA. The pre-mRNA is the original one that comes off. And then you'll notice that this one, the mRNA, happens after RNA processing, and it's a little bit shorter. It's a little bit shorter than the other one. So what happens there? Why is that? Well, we'll take a look in just a second. But notice in the bacterial cell, DNA to mRNA, no editing, okay? No editing, no processing whatsoever. Eukaryotic genes have junk. You may have heard of this before. You may have heard of junk DNA. And by the way, it's not really junk. It's just some codes for some things, but the introns are what we call the junk, and they're learning that more and more some of those differences in the introns are there for reasons, and they also give us some of our own specific differences between people. There's things called single nucleotide polymorphisms, which are called SNPs, SNPs, and they maybe would account for a lot of our unique differences. So they're still looking into all that. So we have exons and introns, right? And the way I like to have you remember that is exons get expressed, okay? They are going to express the gene, meaning this is the code we need to make the protein, okay? Introns are in between the sequence. Introns are in between, and they're the stuff we don't necessarily need to make a particular protein. So introns must come out, okay? Introns get spliced out. So if we look at our diagram again down here at the bottom, the exons are going to be the blue segments, with the introns being those orangish colored ones. So we move to the next slide, and we have the same kind of colors there. And with our post-transcriptional processing, after transcription, we're going to do some editing, okay? And what this is called is splicing, okay? We're going to be doing some splicing because we have to edit out the introns, okay, to make our mature mRNA transcript. We're going to go from the pre-mRNA to the mature mRNA. And so the introns are the orange ones, and we have to be able to cut these out. And so we're going to go, hold on a second. We're going to work through all of the splicing, and we're going to go from about 10,000 bases down to about 1,000. And this one down here at the bottom, if I quit moving my mouse and that picture goes away, you can see it's much shorter. And now it also has all of the green pieces put together. And I'll tell you why they're green here. Actually, I think there's some labels. There we go. Forgot to put the labels up. So there we go. So the reason, so up here we have blue, and that's just showing you that this is DNA, and that these become green because now we're talking about a different sequence, right? We have the U's in there, and we have our primary mRNA. So the green ones are the exons now. We still want to cut out the orange ones, and once we do, and we cut out those orange ones, then we're left with only the green parts, which are really the parts we want, and we have the mature mRNA transcript. Okay, so the DNA makes the original mRNA, and then that is going to get edited, and we're going to take out all the parts we don't need, and we're just going to be left with the sequence that we need to make the protein. Splicing has to be accurate, no room for mistakes. So if we cut wrong, if we splice out an intron incorrectly, we're going to end up with all sorts of problems, right? So here's our original sequence, right? Okay. And so when we do some cutting here, we are going to then have our RNA in here, and notice that this stuff was cut out, the red was cut out, and we have our two G's right here that we're paying attention to. And so if we look, if you remember, things get grouped together in threes, right, called codons. And so here's a codon, and here's our two blue G's, and then all these others make codons, right? And ultimately those code for these crazy abbreviations down here, which are amino acids. And we will get to all of this tomorrow, I promise, in more detail. But just so you know, what happens if you wouldn't cut this properly? Here's our two blue G's right here. Well, what if we didn't splice this properly and we got one of those red G's in there on accident? And so now we have three letters in here when it should be like this put together up here. Instead of getting CGG, which is what we wanted, now we have CGG, but now the blue one is over here. Instead of UCC, we got GUC. And instead of GAU, now we got CGA. And so you look, all these pairs are now wrong. All these groups are wrong because we inserted a letter by accident because we did not splice correctly. So if we cut in an extra letter or we miss one and we cut one out, that's going to shift everything around. And now all the amino acids are different. And you might actually end up with a stop codon. You can see the word stop down there. And now, do you think the same protein will get made? Well, no, because the one on top in the blue box, that's our regular one, we should still be adding amino acids, right? We should still be building this protein, but the one down below stopped. So you're not going to get what you are supposed to as far as how a protein gets made. And then it will probably give you some sort of disorder or disease, okay? So here's how the splicing really works. And there's some really funky words here. One of them being the spliceosome, okay? We have the spliceosome, and then we also have SNRPs, okay? So I think we can put them up here. SNRPs, small nuclear RNA and protein, and then spliceosome are several SNRPs. Okay, so let me get all that up there. So SNRPs, so you have nuclear RNA and protein. Those are the things that are going to be the doers. They're going to come together and be the little molecular scissors to do the cutting, to do the splicing. And then a spliceosome is several SNRPs together. They're going to recognize the sequence that they need, and then it basically becomes a cut and paste process. Okay, so if we look up here, here's our orange intron that we want to cut out, right? So you have some SNRPs here and some SNRPs here that are going to bind to those sequences, and notice they kind of come together and they're pushing this part of it this way and this part of it this way, so that they're bringing the two green pieces, the exons, they're now bringing closer together with the loop of the part you don't want up top. And that loop, I'm sorry that it's these letters are one off, I'm not sure why it's doing it, but it's called a lariat structure. That T should be right there. Okay, it's called a lariat structure. And so when the SNRPs bring these two things together and this intron is sitting up here like this is where the cut's going to happen. And you're going to cut out that intron, and I'll let the cursor wait for a minute, and you can see at the bottom, now we have those two exons pushed together, and we have a separate circle, what used to be the intron, and it's been excised, or it's been cut out, okay, by the SNRPs and the spliceosome. So if we look, and I move my head, we have what's called alternative splicing, alternative mRNAs produced from the same gene, okay, from the same gene. So there's several exons in a gene. It can be spliced many different ways. So all these colored boxes right here are all exons, right? And all the light blue are the introns. Well, look, we can splice these a lot of different ways. So we can splice the green and the orange one in if we want to. Or if I get rid of that and we look at the bottom one down there, we could splice in just the green one or just the orange one. So do you think this and this will give the same protein or not? Well, one of them has the sequence with the green in it, one of them has a sequence with the orange. So you're probably going to make a different protein, and we made them from the same gene. Didn't we say earlier that it was one gene, one enzyme? Didn't a couple guys win the Nobel Prize for that? Beadle and Tatum? Yeah, they won the Nobel Prize for one gene, one enzyme. What did we just show? We just showed that you can make more than one protein from the same gene. So they just didn't know about alternative splicing at the time. Nobody did. That this was possible. So you can actually make many different proteins can be made from the same gene. So remember, they used to think there were 100,000 genes in the genome, and there ended up only being around 20,000, give or take. Well, it's because you could splice them in a million different ways. So they knew there were more proteins than 20,000, but everybody was just then figuring out all the alternative splicing. And so now you can make lots of combinations from the same gene, and it all has to do with this editing. It has to do with how they get spliced together. A little bit more on post-transcriptional processing or editing. We have to protect this mRNA as best we can, because if you remember, DNA never leaves the nucleus, right? The cytoplasm is a very bad neighborhood, and you do not want the DNA that has your code. It has everything you need to build the molecules that you need to live. You want to protect that, so the DNA is going to stay inside the nucleus, and we're going to send mRNA out into the cytoplasm, out to the bad neighborhood for us. Well, before we do that, enzymes in the cytoplasm will attack the mRNA. That's why it's a bad neighborhood. So we have to protect it as best we can. And one way we do that is we put a 5' GTP cap, which is a modified nucleotide, and we do that at the 5' end. So at the 5' end, this G cap, it's not a regular G, it's a guanine or triphosphate, right? Remember ATP, we have GTPs, right? We have guanosine triphosphate, but this G is funky. It actually gets put on kind of upside down. And so the structure of the GTP cap makes it near impossible for anything to latch onto it. They don't recognize the geometry of how that G works, and so they have a hard time getting onto it. If they could bind to it, they would bind to it and they would just start chewing it up, and that would be bad. So the GTP cap protects the 5' end, and then we add what's called a poly A tail to the other end. And so the longer the tail, the longer the mRNA lasts, and more protein gets produced. So we just keep adding a whole lot of A's, a whole lot of adenines to the 3' end, somewhere between 50 and 250 A's, so that even though some of these enzymes will recognize the adenine and they will attach and they will start chomping them down, we don't care if we lose 25, 30, 40, 50 bases. It doesn't matter, because the information we're really trying to protect is in here. We don't care about the tail. Okay, all it has to do is last long enough for it to be translated at the ribosome, and then our protein's going to get made. So the longer the tail, the more this mRNA might stay active, and then you can make more protein, okay? Whoops, let me get rid of that. So that's really the story, right? DNA. So DNA, just wanted to put the whole story up there. DNA is the picture here is that we keep DNA in the vault, right? And these are our blueprints that our bodies need. And in order to get the proteins made, we'll make copies of them, right? We'll make copies of the blueprints in the form of mRNA. And then that mRNA will have to leave and it will have to go find a ribosome, which is the protein factory, right? And then we're going to make some proteins. We're going to string all the amino acids together, make some proteins, which is this guy, and that's ultimately going to give us the trait or the phenotype, right? Which is meaning your protein or your enzyme works or it doesn't, okay? So that's the first part of the process. That's how transcription goes, how we make mRNAs and how we edit them to give us all of these different options to make tons of different proteins. So hopefully you guys understand that a little bit better, and we'll talk to you soon.